it's time to begin. Today, I'd like to extend a warm welcome to all of you for joining our webinar hosted by CellarisBio. Today's fascinating presentation will provide insight into practices of target discovery and validation, together with importance of target engagement in designing winning drug discovery programs. We have two highly knowledgeable speakers on the panel today. And first speaking will be Dr. Chris Miller, PhD, and we'll also have Dr. Elmar Nurmemmedov. I'd like to present our guest speaker, our star of the show, Dr. Miller. He holds a PhD in pharmacology. He is an ex-head genomics research center at Abbvie and ex-VP of functional genomics at GSK. Chris is passionate about technologies to determine function of genes and genomes, identification and validation of drug targets, as well as translating results from human genetics into drug discovery programs. Then our second speaker will be Elmar Nurmemmedov, PhD and MBA. Dr. Nurmemmedov is a co-founder at CellarisBio. He holds PhD in molecular biophysics, trained at Harvard Medical School and Scripps Research Institute, later principal investigator at St. John's Cancer Institute. His lab focused on in vitro pharmacology and early drug discovery. Elmar holds patents for small molecule therapeutics and is inventor of several novel drug screening technologies. So it will be a very informative program today, as you can imagine. Just to let you know, after our speaker presentations, we will have some time set aside for Q&A session. So please note at the bottom of your screen, there's a button, Q&A. Feel free to use that function throughout the webinar to submit your questions. And at the end, our professionals will be happy to answer those questions. So now I would like to welcome our first speaker today, Dr. Chris Miller. We are ready to hear what you have to share with us.
Great, thank you. Do I have control?
You have control. Go ahead.
Brilliant. Okay, thank you. So I'm going to start out with some historical perspective on the pharmaceutical industry as a whole, and some of the challenges that the industry has faced and continues to face. And so let me just dive right in. So these two figures are from two separate papers that came out about a decade ago, but they were caught a lot of people's attention. And the one on the left is a industry-wide analysis of cost per drug launch. And basically what you can see is a decrease over time, which is not good. I might have not said that. This is drugs per billion spend. So you actually don't want this number to go down. You don't want drugs per $1 billion to go down. You want it to go up. It's certainly not down. And over time, this is actually depressingly trending in the same direction. And so this caught a lot of people's attention and caused a lot of hand-wringing throughout the industry. On the right is a paper that actually is, I consider to be required reading for anybody in pharmaceutical R&D. It was a very thorough breakdown of all stages of preclinical and clinical drug discovery. And for each of those stages, the probability of technical success, the number of works in progress needed to get one launch at the end, cost, timelines, and really a very nice breakdown of drug discovery and drug development. And it also painted a pretty sobering picture. A couple things to draw your attention to. One is that at the time in 2010, it was $1.778 billion per launch. And that's a hell of a lot of money. And also one thing to focus on is most of the failures happened in phase 2. And this is a critical stage of drug discovery, drug development, where candidate drugs or lead candidate drug molecules are tested in human patients with the disease that's trying to be treated. So very expensive, and phase 2 is the problem. Wind forward a few more years, and there's reasons to be hopeful and reasons to not be hopeful. So on the left is there's many papers that analyze success rates and trends in the pharmaceutical industry in R&D. And this is a recent one from 2020. And what it shows is that the number of approved drugs at FDA has actually shown an increasing trend. I'm not sure what happened in 2016 where it dips back down, but the number of approved medications is higher now than it used to be. So that's good. However, the average and median peak sales per new drug are no higher. In fact, they are lower now than they used to be. And the total revenues from these approved drugs is really no better. So we're making and getting more drugs approved, but they're not making more money. On the right-hand side, another recent paper that really sends a profound message, a couple profound messages. This is the R&D efficiency. So this is the amount of money that is spent for each new medical entity. And on the x-axis, it's how much each new drug makes. And ideally, you want to be certainly on this side of the diagonal line that would at least make it so that the companies were making more than they spent on individual drugs. But what's sobering and disturbing is that most companies, many companies, are in the upper left-hand corner of this graph, where the amount of money spent per drug, per marketed drug, is anywhere between $4 and $8 billion. So I showed you on the last slide that it was 1.8, and that caused a lot of concern, but it's now much higher than that. So many companies are in this red zone or upper left-hand corner, and they're spending much more money on per drug than they're recovering. Now, this paper gets into some nuances of what was developed internally versus what was in-licensed or acquired through mergers. So it's not quite that simple. Takeda apparently is the only major pharma company who couldn't rescue their way out of a poor pipeline by mergers and acquisitions. So even though some of these other companies are in this red zone, they've been able to make up some of the gap by acquisitions and mergers. Anyway, so I'm just going to talk about a few things that the pharmaceutical industry has done and is doing to try to increase the success rates, decrease timelines, bring down costs, and so on and so forth. This is not meant to be an all-inclusive list, but I'll talk about some that I'm more familiar with. Human genetics, functional genomics, artificial intelligence, decision-making, and better pharmacology. And these are places where target engagement that Elmar is going to talk about can have more of an impact than they currently are. There's other things like translational R&D, new drug modalities, external innovation, academic partnerships, chemogenomics, phenotypic screening, systems biology, and so on. Many things that the pharma industry is doing, but in some cases they're flavor of the month, flash in the pan. Others, they're more substantial. So this is not meant to be a complete overview, just commenting on a few things that I'm familiar with. So back in the mid-2010s, two papers in particular caught a lot of attention of the industry, one from AstraZeneca, one from GSK. And the take-home point from both of them is that human genetic support, if you have it, it can play a huge difference in the success rates of drug discovery and drug development programs. And so on the left-hand side is the AstraZeneca paper, and this is indicating yes or no to the human genetic support, and blue is active or successful, beige is closed. And what you can see is when you do have this genetics support, 73% of the total programs in phase 2 were active or successful, whereas when you don't have it, almost 60% of them were not successful. So strong correlative information showing that human genetics data matters. On the right-hand side, a similar sort of message from the GSK group, basically saying you have human genetic support, you're going to do better in drug discovery and drug development. And they quantitated it to say actually twofold higher when you had genetic support. So every large pharma company has put big effort on human genetics to build it integrate it into the target discovery, drug development pipelines. Functional genomics, again, something I spent basically all of my career in the field of functional genomics. And without going too far into this, I mean, I could spend a whole hour talking about functional genomics and drug discovery, but it refers to the combined set of technologies and analytical methods to determine the function of genes and genomes and other omes. So you hear about genomics, proteomics, metabolomics, any genomics, transcriptomics, and so on. So it's the idea that there are technologies that have been rapidly developed that allow us to measure at scale, to perturb genomes at scale, to analyze at scale, to do everything in large scale functional biology. And functional genomics impacts multiple stages of drug discovery and drug development, most commonly initially used as a way of discovering new targets, but it can do many other things across the pipeline as well. And in my times at Wyeth, Bristol Myers Squibb, Abbvie, and GSK, we had many examples of doing all these things across the pipeline. So it's not just about targets. It is about targets, but it's not just about targets. I'd be remiss if I didn't include a few comments about AI as another transformational approach to drug discovery and drug development. And we were among the early adopters in these approaches while I was at GSK. And I won't go through this in a lot of detail, but there are many ways that AI is starting to impact different aspects of drug discovery and drug development. Drug design, drug interactions, chemical synthesis, drug repurposing, drug screening, and it's many, this is drug discovery, many things in drug development as well. So patient selection, biomarker strategies, imaging technologies. AI is starting to and is going to continue to transform drug discovery and drug development. Again, I could spend a whole hour talking about ways that this is happening and why it's important and what's exciting about the future. So more operationally, there have been several papers that were published, this one in particular from AstraZeneca, that this is actually this is a 2018 follow-up to a paper that was published in, come to it later, I think it was 2012. And the idea is that it used to be that it's not that it was the Wild West in drug discovery and drug development, but there weren't sort of standardized frameworks for thinking about it that were used across the industry. And this is a paper that was summarizing the impact of this 5-dimensional framework. And it's the 5 R's, or right target, right tissue, right safety, right patient, right commercial potential. And in truth, there's another R, right culture, that they refer to later, but didn't sort of make it as a standalone part of their framework. But this was actually a paper that caught a lot of attention. I guess I said that several times, and hopefully the papers that I am referring to here were meaningful. They were meaningful to me and the companies that I was part of, and I think the industry paid attention to them. On the right is one particular graph from that, one particular figure from the paper. And I showed this one because what they did is looked back across the AstraZeneca pipeline at many metrics associated with the attributes of the 5 pillars that are shown on the 5 R's that are shown on the left. And one in particular I'm highlighting here is this is outcome of projects with or without proof of mechanism. And basically, if you don't have good data showing proof of mechanism, the projects get terminated. This is once they hit the clinic, whether they get to phase 2 or phase 3. But most close, when you don't have proof of mechanism, most projects don't make it into phase 2. And the proof of mechanism is important in the context of this presentation, because target engagement is a key piece of demonstrating proof of mechanism. Here we go, sorry. Another paper that caught a lot of attention, this came out while I was still at Abbvie, but it was from Pfizer, and it's basically making a simple point that many programs in the pharmaceutical industry were pushed forward without having data addressing what they call the 3 pillars of survival. And so pillar 1, this is basic pharmacology. Pillar 1 is exposure at the target site of action. Does the drug get to the site that you think it does to elicit its its mode of action? Pillar 2 is, does it bind to the pharmacological target that you think it does? And this is target engagement. Does it bind to the target that you think it does in a relevant cell type or biological sample? And then third is, does it cause pharmacological changes that are consistent with the mechanism of action? So these 3 pillars of survival, what Pfizer did nicely was they went back and looked at their portfolio, and I'd not shown on this slide, but there's a corresponding table, graph, 2x2 graph of what their pipeline looked like in terms of how many of their programs had both pharmacological confidence and exposure confidence as contributed by the pillars, and how many only had one and how many had neither. And the long and short of it is that those that had high pharmacology confidence and high exposure confidence did much better. They made it into phase 2, made it into phase 3. They didn't include data on whether they were approved or not, but they made it further along. And so it's just underscoring that if you don't pay attention to basic pharmacology, the programs are not going to advance, or if they do advance, they're going to not be successful. So a couple slides on target engagement, and really I'm going to just go through these quickly so that Elmar can spend the time. He's the expert on target engagement. But a couple of just general points I want to make is it is becoming, there are increasing number of papers in the literature, not a large number by any means, but people starting to recognize the importance of target engagement and making suggestions on the best way to use it. And this is a nice paper from Eli Lilly that basically just made the case for having target engagement be an integral part of your early drug discovery activities, not just something that's done on demand or as needed, but it's an integral part of it. And I'm a little bit surprised that the case has to be made for it, but apparently it does. The paper on the right was a really nice short review article, just making the case again for target engagement, in particular in cells. Many of the early target engagement technologies were cell-free, were done on outside of cells, on lysates or purified samples from cells. And being able to do the target engagement in cells is important, because things change when you bust up cells and make lysates. And being able to show things in an intact cellular environment is important. This is just this is an old slide that really, I just was trying to tie things together and make the point that target engagement can be applied and should be applied in hit to lead or lead optimization, but it can be applied. You could think about doing target engagement type of assays as high throughput screens or medium throughput screens. And the information that you generate from target engagement will impact every step of the pipeline, whether it's preclinical or clinical. And just this is my last slide. You know, where the industry gets new targets is critical for pipeline sustainability, probably stating the obvious. And they're going to come from a number of sources, whether they're from the literature, functional genomics, omics and multi-omics profiling, systems biology, data mining and artificial intelligence, human genetics, phenotypic screening, and probably others that I haven't listed here, but they come from a variety of sources. And it's important to have eyes wide open for all these sources of targets, because every pharma company is doing that, and it's a matter of being competitive, but also identifying opportunities before others do. And just some of the challenges, good targets are becoming harder to find. The definition of druggability is evolving. That's actually an opportunity in addition to it being a challenge. Druggable definitions of the druggable genome continue to evolve, and new targets and new target classes are being identified. There's often in uncharted territory in understanding the biology and disease. The models are not very translatable, whether it's in animal models or cell models. Limitations in access to many human tissues and clinical samples, and then differences across disease areas. It's not a one-size-fits-all. There are the approaches that work and the sources of targets in cancer are very different from the approaches that work and the sources of targets for metabolic diseases, or it's different for every disease area. So again, you could view these as challenges or opportunities, and some of them may be both. So I'm going to stop there and hand it over to Elmar. Or Elmar, you can take control of the screen.
Thank you so much, Dr. Miller. Fantastic presentation. Thank you for sharing your knowledge with us. And before we pass on to Dr. Nurmemmedov, I'd just like to remind everyone that we have the Q&A available below for you to put in your questions, and they'll be answered at the end. Now we'd love to hear from you, Dr. Nurmemmedov. Thank you.
Thank you so much, Chris. Thank you, Brooke. So I would like to make a few points about designing new drug discovery programs, especially with the emphasis on challenging drug targets and cell target engagement. Now, there is one dilemma with modern drug discovery, and this is becoming a really good picture that we have to face. So disrupted cell biology is one aspect of current drug discovery. Conventionally, there are more than 20 methods that we employ in early drug discovery. These could be recombinant production of the protein, the target protein, the multiple biophysical methods, the disconnected data that we have to stitch together in order to make sense of the early stage in vitro pharmacology. And in many cases, the disrupted cell biology essentially might be somewhat misleading in those efforts. Challenging drug targets is another layer of challenge. There is opportunity here, but challenging drug targets command another way of looking at drug discovery altogether. These are targets that I will mention in a separate slide that essentially are not easily druggable using the conventional early drug discovery methods and tools. And also there are more than 9,000 undiscovered therapies out there. And many of these undiscovered therapies essentially are locked behind challenging drug targets. Now, in terms of biotech and pharma, there is a great opportunity here to really unlock these new therapies. And there is also economical and clinical benefit there. Now, target selection is key in drug discovery. That's the first thing you do, right? So when you go for target shopping, you need to make it very clear, is that the cancer that you're going after, is that the neurological disorders, metabolic diseases, or any other essentially disease states. And this will impact the economics of your drug discovery and the pharmacology and essentially everything that's related to your drug discovery programs. Just to give you some numbers, there are more than 30,000 genes in the human genome. You know, the number can vary if you consider multiple splice variants. Only 3,000 of them are considered as disease-modifying genes. So essentially genes that have the potential to impact, the state of the disease. Out of that, there are, just around 1,000 drug targets. The proteins are essential that are, druggable, undruggable, essentially, that have the potential to reverse the course of the disease if you hit them with the proper therapeutic modality. And it's interesting that 80% of the drug targets are still considered challenging or undruggable, if you will. And this is despite of the fact that the drug discovery is expanding with its arsenal of multiple therapeutic modalities and multiple ways to approach the targets inside or outside of the cell. Another look at early stage drug discovery and what target engagement can do. Essentially, on the left side here, it's your genomics, proteomics data that will dictate and that will shape your drug discovery. Once the data is in, it will go into design stage with target ID assay development, then discover stage, screening for small molecules for hits, and essentially you will identify your hit and try to optimize it under lead optimization. And essentially then you go into advanced preclinical work, mouse work, or invertebrate work, and then you go into clinical studies starting on phase 1. This is a very effortsome procedure. Conventionally takes upwards of $700 million just for the early stage. If you consider the entire drug discovery development journey, this is one of the most expensive elements and sections of that journey. So the big questions are, is your drug candidate really therapeutic? Does it engage the target inside the cell? And it's rather important to answer these questions early in drug discovery rather than leaving them to the end. If you look at the failures in drug discovery in the clinic, you will see that in phase 1, it's pretty much equally efficacy and safety of the drug. Phase 2, the failures are mostly related to the efficacy of the drug. Essentially, does it hit the target? Does it do enough of clinical benefit? And in phase 2, it's also important to emphasize, the efficacy and, of course, the commercial viability at that point. So a lot of these efficacy and safety-related concerns can be addressed early on in the drug discovery to really mitigate that risk in the clinic, to make your drug discovery program more efficient and de-risk it sufficiently for its commercial viability. So high risk of failure due to poor drug target engagement can be addressed at early stages of drug discovery. Now, there are various ways to discover a drug in early stages. For example, if you start with a phenotypic screen, you have an elegant cell-based system, and you have a library that you want to screen, and then you have your readouts, either those are phenotypic morphological readouts or some kind of biomarkers, and you will essentially find those best compounds after sufficient screening. But the question will be, what is the target? You may have a good response, but what is the target? Second, you may have a target-based screening. Essentially, you have a recombinant version of your target protein expressed, purified in whatever host, and then you have your library. You can go through conventional or advanced screening procedures through reporter assays, and eventually you will identify that potential hit compound or your best candidate. Then the question is, does it really engage the target inside the cell? Because the assay has been done on a recombinant version of that target. Now, most recently, AI and machine learning is really driving drug discovery. Structure-based drug design is one side of that. Essentially, you can in silico or computationally design a drug discovery effort. You can go through multiple phases, eventually plug in some orthogonal assays using purified or recombinant version of the target. Eventually, you'll find that best hit compound. The question is, does it really hit the target inside the cell? So really, it comes down to that main question. Does your candidate engage the target inside the cell? And this becomes a holy grail for early drug discovery. Now, I want to put an emphasis on challenging drug targets, because challenging drug targets are that 80% of the untapped 80% of the human proteome. So what are challenging drug targets? Why are these so important in drug discovery? And why are they value as for future drug discovery programs? Now, on the left side, you have druggable conventional drug targets. These are enzymes with well-defined active sites. You fit a small molecule. Essentially, you inhibit the function of the enzyme. Transporters, these could be various membrane proteins that, lead across various types of ions and analytes. Receptors such as nuclear receptors. These are very important proteins that essentially regulate some hormonal functions and other biological functions. And some GPCRs, also known as transmembrane proteins, G-coupled protein receptors, important functions to these proteins. These are druggable targets with conventional means. And in fact, most of that drug discovery up to date has been utilizing and exploiting these multiple targets. On the right side, now we have challenging drug targets. Now, challenging drug targets have multiple interesting features to them. First, lack of hydrophobic pockets. Essentially, the target does not have an active site, and it may have very shallow or crevices here and there, but there is no defined site where you can fit a small molecule. They function via protein-protein interactions. So if you hit just the target itself, may not be significant. You really have to hit the interface where it has that significant disease-modifying function. Active sites are highly conserved among family members. If there is an active site, like in the case of KRAS, maybe that active site is highly conserved among other family members, leading to unnecessary, undesired cell toxicity. And very importantly, lack of three-dimensional structure outside of the cell, and impossible to synthesize the protein recombinantly. Because there is no structure function for these challenging drugs outside of cell, essentially it becomes impossible to produce that target in a sufficient form for an efficient drug discovery program. These are challenging drug targets. And it's important now to underline that challenging drug targets inside the cell have multiple functional or structural states. Now, on the left side, you see what conventional biophysics or simplified biophysics will teach us. There are many energetic states that a protein can assume inside the cell, but we tend to know that the one structure assumed by the target protein is the state of lowest energetic basically level, and the protein will stay at that level and essentially assume its function inside the cell. But that is not actually true. Inside this cell, a challenging drug target may actually undergo various structural transitions. It can assume one structure depending on one protein-protein interaction, and it will then transition to another more energetically favorable structure if it goes and becomes part of another protein-protein complex. So essentially, it's important to appreciate that these are not proteins with one defined structure, but most of them are intrinsically disordered proteins that can assume various conformational states. And so this here graph represents that between the lower energy conformation and higher energy conformation inside the cell, the challenging drug targets can assume actually a mixed population of states. Now, a few of those states might be more predominant than the other ones, but it's important to understand that there are multiple conformational states to the proteins. And then the question becomes, does your candidate engage the right conformational state inside the cell? It's very important if you're going to design powerful and efficient drug discovery programs. Now, there are ways to approach challenging drug targets in an agnostic way. What I mean with agnostic is that we don't want to be locked into their one particular structural element, but agnostic in a way that, we want to develop and discover therapeutic modalities that will hit these targets inside the cell, whatever their conformational states and whatever their structural elements. So the problem is, again, the problem here on the right side, on the left side, is that challenging drug targets do not have a well-defined binding pocket, or the pocket is too shallow, or the protein is a part of a protein-protein interaction. And now the desired solution is, for these type of targets, is to discover a hit compound or a modality that is large enough to span a significant amount of surface area on the target, such that it will interfere with the function or interfere with the protein-protein interaction that target is involved in. Now, one way to approach this is through cell target engagement. Now, cell target engagement provides you the agnostic approach to these challenging drug targets. You could design a reporter system, a reporter cell, using whatever disease-relevant cell model that you have handy. And for any specific drug target, you can design a reporter. You can design now a completely new drug discovery effort. One way to do that is to pass through your cells at various thermal levels. For example, temperature of aggregation 25, 50, and 75. And you can discover small molecules or modalities at the thermal edge of your target protein. If a modality, a candidate, or binders engage with the target inside the cell, they will thermally stabilize the target, and you can essentially identify them and pass through your other orthogonal assays. So cell target engagement becomes a very important integral component of early drug discovery, especially for targets that do not have a structure functional side of the cell. How does cell target engagement work? Now, you can imagine that inside the cell, your targets are essentially in a fluent structural states. When a ligand binds, engages the target inside the cell, the target goes through some biophysical transition and becomes more thermally stable. If you put that target through a temperature gradient, essentially what will happen is that the unbound target will follow the blue line. Essentially, it will melt, and you will see that the temperature of aggregation is could be at 45, 47 degrees Celsius, right? Usually it's a lower degree. When there is a ligand that is bound to the target, the target will undergo thermal stabilization. It will still melt, but it will do so at higher elevated temperatures. So there is that thermal stabilization event that will happen upon engagement with the target inside the cell. And we can utilize this for drug discovery purposes. The advantages of this is that cell target engagement is done under physiological conditions of the cell, relies on thermal melting of the target protein, no modification or minimal modification of the target, no modification of the compound or the therapeutic modality you're testing, disease relevant, and you can design, multiple applications, including discovery, validation, analysis of drug candidates. Now at CellarisBio, we have developed a microtag cell target engagement platform. This is a multifaceted platform that allows you to interrogate multiple targets and allows you to do multiple things, multiple efforts with those targets. The first one is discovery of the chemical matter. So we can work with potentially any type of drug target. We can work with membrane proteins, GPCRs, transcription factors, enzymes, and you name it. We can discover the chemical matter using DELs or conventional plated libraries. Once we identify the chemical matter, we can go through actually a validation step using quantitative cell target engagement. We can measure the EC50 of potency, and we can also rank them by selectivity, and we can identify the compounds that actually are strong and selective enough. once we identify the desired compounds, we can now use real-time cell target engagement to really understand the mechanistic details and to gain more insight into their engagement patterns inside the cell. And altogether, the platform allows now us to really identify the targets, validate, discover and validate hits to those targets, and then really in an efficient way push those best candidates into preclinical work. A little bit about the chemistry of the science behind cell target engagement system. Now, inside the cell, the target can be anywhere in the nucleus, in the cytoplasm, on the membrane. The first thing is if you clonally attach a 15 amino acid tag to the N or C terminus of your target protein, introduce that into the cell. Now you have an engineered reporter cell for that specific target. You will treat your cells with your ligands. You will expose the cells to the temperature challenge, which is a unique temperature for that specific target to really thermally perturb the target. And you would then detect the signal by adding the FRET substrate and attack binding protein. You would then choose what you want to measure. You can use conventional plate readers to read the end point of that signal, accumulated end point. You could go into real-time readers where you can actually see the entire sequence of events and monitor the entire engagement event from time 0 to time X. Or you can go into real-time cell imaging systems where you can visualize the target abundance in terms of degradation or target accumulation. So Microtech cell target engagement system allows you to really bring your drug discovery efforts to a whole new level. The enabling workflow of the system can be simplified to 4 steps. First is you select the target of interest. Again, we can work with very challenging drug targets. Second, we will engineer a microtag reporter cell for that specific target. Of course, there is some optimization. There is some pilot tests that we have to run to make sure that it responds and the assay is sensitive enough. Step number 3, then go ahead and do cell target engagement system. You can do discovery, validation, optimization, and real-time analysis. And this will essentially complement a lot of those other assays you might otherwise do in your lab. There is going to be some medicinal chemistry involved here, and this step number 3 can actually go in multiple cycles, iterations, until you find that potential compound. And then you fast forward, you can go into preclinical studies with much more confidence, knowing that you have a best candidate that engages the target in the cell with potency and selectivity and without unnecessary cell toxicities. And these will give you the answers, and after which you can proceed to the next phase of your drug discovery program. I would like to present you one case study we have recently worked with. This is a GPCR protein X that we worked with. As you know, GPCRs are superfamily of membrane proteins, and human genome encodes approximately 800 of these proteins. Upon ligand binding, GPCRs undergo a conformational change. Now, there is a stabilization event, and this is actually an advantage as you exploit these targets for drug discovery programs. Now, when we started working with the GPCR-X, it was a very challenging target. And in fact, the conventional tool compounds did not respond and did nothing to GPCR. What we decided to use is a real-time cell target engagement. Real-time cell target engagement system that we have developed in our lab allows you to really scan various temperatures. For example, in this case, from 45 degrees to 65, 70 degrees, and expose the target to various temperatures and see at which point the target will respond to the temperature, and at which point potential ligands will be able to stabilize or destabilize the target. This is an agnostic approach. Now, the conventional tool compounds that we had available for this specific target potentially tested at various doses did not give us any measurable effect. As you can see that when we proceed from 40, 45 degrees Celsius all the way to 70 degrees Celsius, we do not see a separation between the control and lower and higher doses of that drug. So there is no thermal stabilization. We tested another tool compound, which is a commercially available compound to this particular class of compounds. And again, there is no response. There is even if you compare the lower to the highest temperatures and controls versus the lowest and the higher doses of that drug, essentially there is no stabilization event. What we did next is we tried a pool of promiscuous compounds. So now we have various compounds that we can use in individual or in a pooled fashion to interrogate compounds. And these compounds will bind to potentially multiple sites across on the surface of the target protein. And this actually opened up the target for drug discovery. So if you consider, if you think about it, starting at 40 degrees Celsius, going all the way to 70 degrees, the DMSO control and the lower doses of these drugs essentially will give you a pattern, a low basal signal. And as you increase the treatment to 1 and 5 micromolar, this red and black line, you will see that this signal that it generates is much more substantial, and you will have a lot more signal originating from these doses, indicating that, first of all, it indicates that the temperature of aggregation of this protein is actually around 45 degrees. So 45 degrees is the temperature at which these compounds will have a thermal stabilization on this target. And we can also see that there is potentially another melting event happening between just around 50 degrees, suggesting that this target may be undergoing multiple states, conformational states, and maybe there is an opportunity to identify some cryptic allosteric pockets on the surface of this target protein. So my take-home message with this case study is that cell target engagement will provide you an opportunity to design assays that will give an unbiased approach to otherwise challenging drug targets, interrogate them in such a way that other assays may not necessarily reveal their druggability. So in this case, GPCRX is now a druggable target in our system, and now we can safely and confidently proceed with testing other compounds to this target. So this is my last slide. Essentially, you can design a first-in-class programs on challenging drug targets. The conventional path will take you 5 to 7 years. With unclear cell target engagement, you will need additional validation and screening, and you will have to do complex multi-parameter optimization of your data, and eventually you will get a drug candidate. It's going to take you a much longer time. With cell target engagement, we're looking into 1 to 2 years. There is a lot of clarity with cell target engagement. No further validation may be needed once you have that data confidence, and there is a path with a rapid hit to candidate generation here. And eventually you'll get a drug candidate in a much more efficient and de-risked manner. And cell target engagement will essentially, de-risk and accelerate early drug discovery. Brooke, back to you.
Thank you so much, Elmar. Thank you both for that fascinating presentation. Now, we'd like to get your participation and get your feedback from everyone. So we're going to launch a poll on the screen. You should see it come up on your screen. The question is, are you interested in testing CellarisBio's cell target engagement technology? Please give us your answer. We really appreciate it. We'll give you a few minutes to answer there. Also, thank you for your participation with your questions in the Q&A. We have a little list there to answer. So we'll get to that right now as soon as the poll votes come in. Thank you so much. Now, some of the questions that we have here We have 4 questions. So Elmar and Chris, we'll throw these questions out at you and see who has the answer. First of all, we have a question about the emphasis on discovering and developing pan-tumor biomarker targets. The question is there still an emphasis on this, or has emphasis turned back to specific disease due to heterogeneity of disease-specific MOA and or cost of time and efforts to fulfill cohort size needs for approval? Would one of you like to answer that question?
I'm not sure who it's directed at. In truth, I did not spend my career inside oncology drug discovery organizations. I interacted with them quite a bit. But to me, this seems like it's a choice between going pan-tumor, like looking for drugs that could impact broad arrays of tumor types, or but to me, the personalized medicine is the better way to go, because it's hard enough to understand why certain people respond to certain drugs and others don't. And getting to the point where things are personalized, where we can predict who's going to respond and who's not going to respond, goes in the opposite direction to the pan-tumor approaches. But I may have misunderstood the question.
All right. Shall we leave that one for now? We can come back to it later. Does that sound good, Elmar? Maybe we can pass to another question. Next one, is drug potency still king? I'm thinking in the context of myeloid neoplasms of PARP1 inhibitors. Who would like to take on that one?
Right. Maybe I can add a few comments. So I agree that potency is very important. In fact, it's the potency that will dictate the most of the success in the clinic. So potency essentially is a combination of, hitting the drug target and essentially giving you the right clinical or therapeutic benefit. So target engagement is very important. It has to hit the target in order to really have that therapeutic effect. So I think if you have the right target in the first place, and I assume that PARP is the right target for the disease indication you're working with, then you have to make that decision. Is there a potent compound that you have in hand that hits the target with the right potency, with the EC50 in the disease relevant model, and that can be absolutely answered using the right cell target engagement systems. And after that, it's the pharmacokinetic, pharmacodynamic features of your compound. You will have to proceed with animal studies to really show that your compound now is not only engaging the target in your in vitro systems, but have the ability to circulate in the system and be in the tissue of interest, and essentially, exert that therapeutic benefit. But I'm not sure if I'm answering the question, but I agree that potency is the right way to really interrogate a lot of drug discovery programs.
I have a question about
Go
the
ahead.
question. Sorry, I can't help myself, but I mean, obviously potency is important, but selectivity is also important. And, I'm thinking about the early days of kinase drug discovery, where it was critical to understand selectivity and specificity. So is potency more important than selectivity, or you need both?
Is that a question for me?
I suppose so.
Okay, so I would say that both of them are equally important. You may be killing the tumor, for example, if it's oncology, but if you are creating unnecessary toxicity, and if, for example, if it's an in vivo study, if the animals die together with the tumor, it's no good. So you wanna you want to essentially keep both potency and selectivity hand in hand. What percentage of emphasis you want to give one or the other, I could not tell. That depends on the clinical program or the drug discovery program, but I would emphasize both of them. And those are the type of questions you can answer early on using the right target engagement assays systems.
Got it. Thank you.
Fascinating. Thank you. We have quite a few questions coming in. We may not have time to answer them all, but Elmar, I don't know if you can see them. Maybe there are some, maybe one more that we could answer. There is one about phenotypic screens, cost and throughput of the TS target engagement system, new targets, repurposing drugs on new targets, thermal stabilization.
So
Great interaction.
is that the question? Can phenotypic screens be used to guide SAR and move drug discovery programs forward? Perhaps I could answer that. So phenotypic screens are really elegant systems to start your drug discovery programs. If you have the right combination of assays, it's fascinating. You will find that great compound eventually doing the right thing phenotypically or morphologically or biomarker wise. So the question is, what is the target inside the cell? And you can answer that question. You can do target deconvolution. Well, there are ways to do that. With mass spectrometry and biotinylation of that ligand that you have discovered, you can fish out that target from this cell. And that's important for you before you go into any type of SAR efforts, because SAR requires you to really optimize the R groups, the moieties on the target to really address the potency while keeping an eye on the engagement. So I would say that identify the target first and then go into SAR. And right now, the tools are available in order to make that program successful.
Fantastic. Any other questions? Please feel free to email us as well. One question I'll leave for you too. Will these slides be shared with the attendees?
I'm certainly fine with it. Elmar, I leave it up to you.
Yes, we will have a recorded version of this webinar. So absolutely. And if you have any questions, please email us and please keep in touch. Happy to answer.
Excellent. Well, our time is up for today. Thank you. And sorry that we weren't able to get to all the questions, but like we mentioned, you can email us at the email that is there on screen, hi@CellarisBio.com. You can follow us on LinkedIn and our website, CellarisBio.com, where you will also find the recording of this program. You'll have access to all the information spoken about today, as well as previous Cellaris Bio webinars. Thank you all so much for attending today's webinar, and a big thank you to our panelists, to Dr. Miller, Dr. Nurmemmedov. Thank you so much. We invite you all to stay tuned for future webinars from CellarisBio. Thank you and have a wonderful day.